Hello, good afternoon. Let's continue with what we were talking about last class, which was the conduction system of the heart. So we learned about a cluster of cells that are located right near the entrance of the superior vena cava. That is called the sinoatrial node, the SA node. So this SA node is capable of generating electrical signals on its own, right? And it's under the control of the autonomic nervous system. So we don't have to consciously think about beating of the heart. The body automatically does this for you, but we can influence the frequency of the SA node. We can influence how fast it's going to be sending out these electrical signals, right? By changing our physical activity, when we have different emotions, or if we have medicine or chemicals in the body, those can influence the rate at which the SA node is firing. So let's take a look at the events that's going to be happening as the SA node send out these electrical signals. So the electrical signals, once created by the SA node, is going to spread across the atria at the top. And these arrows that are that's in the diagram kind of shows you which way the signal is going to be spreading. Okay, so they spread across the atria like that. And as the signal spread across the atria, it's going to cause them to contract. Right. So I'm gonna put the note down here for you. SA node. Okay, the signal is going to spread across. Impulse means the signal. Impulse spreads across the atria. Okay, and at the same time that this is happening, the signal is also going to be traveling to another cluster of cells called the AV node. But we're going to worry about the AV node in a second. Right, let's talk about what's happening with the signal going across the atria. Okay. When the signal reach the atria, we're gonna have atrial contraction. Okay, we'll just say the atria, both of them, okay? The atria contract. And empty blood into the ventricles below. Now, in order for this to happen, in order for the blood to go from the atria to the ventricles, which two valves need to be opened? In order for the blood to go from the atria to the ventricles, which two valves in the heart need to be opened?
The pulmonary and aortic.
Are those the ones that's between the atria and the ventricle, though?
Tricuspid and bicuspid.
That's right. The tricuspid and bicuspid valves open. Okay, they're here, right? Tricuspid and bicuspid valve. You need to open them so that the blood can go down. And we don't want the blood to be escaping from the ventricles before they are completely filled up. And so when the tricuspid and bicuspid valve are open, at this point, the pulmonary and aortic valves are closed. They are open. So ventricles fill up with blood. Right? So all these things are happening when the signal is traveling down to the AV node. Okay. Just gonna give you a moment to copy whatever you need to copy. And, ask me questions. Okay, in the diagram, we have another cluster of cells here. I'm just gonna use a different color for you to see a little bit better. This is the AV node, and which stands for atrial ventricular. Atrial ventricular node, or AV node for short. Yeah, I'm gonna be posting the recording every time, okay? Every time I'm gonna post the recording, and I'm gonna post the notes as well, right? So if you miss anything, yeah. If you don't know where to find the recordings, you can ask me after class as well, or like ask me during the break, okay? I'll show you. So the AV node is the purpose is to relay the signal to the ventricles. Okay. So the signal starts from the SA node and then it spreads to the AV node. Then the AV node kind of boosts it a little bit and pass it on. Think of AV node as like a Wi-Fi extender. Okay, it increased the coverage, essentially. So once the impulse gets to the AV node, the AV node will then pass it on to the next thing. And to keep things simple, all right, like there are extra names for all these stuff, but I'm just gonna, it's already complicated enough, okay? So all these fibers that we have over here, right, and you have more fibers here, okay? These fibers up here, we call them like the AV bundles, atrioventricular bundle. They're just like cables that pass it forward. And then we have more conducting fibers inside the septum, and we call them the Purkinje fibers. Okay. So the signal basically spread down the septum and then up the sides of the ventricular wall. Yeah. So while this is happening, while the signal is being passed down to the ventricles, this whole time the ventricles are filling up with blood. Okay. So AV node goes to the AV bundle and then to the Purkinje fiber inside the septum. Now, when this happens, now it's time for the atria relax. It's finished. They have finished squeezing the blood into the ventricle. They don't need to squeeze anymore. Now it's time for them to relax. And as soon as they relax, they will start to fill up with blood. The atria relax and filled up with blood. From where? Right atrium receives blood from where? Help me out here.
Aorta.
Aorta sends blood to the body, so that's not it, but good try.
Superior vena cava.
That's right. The right atrium receives blood from superior and inferior vena cava. Okay, and then the left side, the left atria, receives blood from pulmonary vein. Okay, so atria is going to be filling up with blood now. What's the ventricles going to do? The ventricles, because the impulse, the electrical signal has reached the ventricles at this point now. So it's time for the ventricles to contract. Right? And empty its chambers. Now, in order for the ventricles to empty itself, as in like to push all the blood forward, it needs to close the AV valves. And the semilunar valves open. Okay, just pause for a second and try to listen to me explain it, okay? Okay, so first of all, at the beginning, you squeeze the atria, the blood comes down, and it fills up the ventricles, right? The ventricles are full now. When the electrical signal gets to the ventricle, we squeeze the ventricles. So we're emptying the ventricles. We don't want the blood to move back to the atria, right? That's why we have to close the AV valves. That's one purpose. The other purpose of closing the AV valve is, don't forget, at this point, the atria are actually filling up with blood. So we don't want to let the blood from the atria to move on to the ventricles before you completely fill up the atria, right? So that's why you need to close the AV valves. But the blood has to go somewhere, right? From the ventricles. That's why we have to open the semilunar. On the right side of the heart, the right ventricle will squeeze the blood through the pulmonary artery so that it's going to go to the lungs. And then on the left side, the left ventricle is going to squeeze and send the blood through the aorta. And that completes what we call like a cardiac cycle. And that's how blood circulates through the heart. Okay, finish copying whatever you need to. If you have questions or if you need me to clarify anything else, please ask.
I have a question.
Sure.
Professor. So, as you said, the tricuspid and bicuspid, they work like their function is when the tricuspid closed and bicuspid open, yes?
Tricuspid and bicuspid, they open
Bicuspid
and close together.
Okay. So, at the same time, the atrium, when it's open, the ventricle closed.
Okay, so when we talk about the atrium and the ventricle, we don't say open or close. For atria and ventricle, they either contract, as in squeeze,
yes, when
or
they
relax.
squeeze,
That's right.
yeah.
So when atria squeeze, ventricle must relax.
Okay, yeah.
And when it's time for the ventricles to squeeze, the atria are gonna relax.
Yes, I got it. Thank you.
Okay?
Yeah.
So for everyone else, right? Like relax means filling up. Right? Contract means emptying. Right? So when atria squeeze, when atria contract, the ventricle relax. That means ventricles are filling up and atria are emptying into them. When it's the second part, then the ventricle will squeeze, emptying the blood either to the lungs or to the body, but then the atria are filling up either from the vena cava or from the pulmonary vein. Okay, you gotta run this scenario through your head a couple of times to get a full understanding of it.
Just to confirm, Vincent, that AV valves are the tricuspid and the bicuspid?
That's right. AV is tricuspid and bicuspid. Yeah, they are located between the atrium and the ventricle, right? That's why they're called AV valves, atrioventricular valves. Okay, everyone else okay? Any other question?
Excuse me, Professor. Professor, SA node and AV node, this topic that today you teach us, it was about the electronic device that put it in the heart.
Oh, no. This is the conduction system of the heart. This is how you generate your own electrical signal so that the heart can beat.
Okay.
Okay, and when it doesn't work, then you need the electrical device. Then you need the pacemaker, right?
Okay, thank you.
Yeah. All right.
One more question,
Sure,
Professor.
sure. Go ahead.
So the AV bundle and
Purkinje fiber.
yes, that what is their function? For example, if
You
we
can think of them as wires, okay? They are
why?
just like help carry the electricity, help carry the electrical signal, basically.
Oh, okay,
Yeah,
okay, yeah.
because you need the signal to get to the ventricles, right? And so that it's gonna squeeze, basically. Okay. So this is what it is, right, in another view, right? You see the SA node up here, you see the AV node, the AV bundle, and then you have the Purkinje fibers. That's basically what it is. Right? And everything I told you about, it's all written here. So a person is gonna need an artificial pacemaker, which is this thing over here, when the conduction system fails. The conduction system can fail in a number of ways, okay? And depending on what kind of failure you have, the result can vary in severity. Okay, some are more serious than others. Okay, so sometimes it can lead to arrhythmia. Arrhythmia is when your heart rate is irregular. Okay, it's not beating in a regular fashion. Okay, like 70 beats per minute. Arrhythmia is like sometimes fast, sometimes slow. Like it's not really um, in a controlled way, right? So your heart is now no longer able to set its own regular pace. And that's why the pacemaker is going to help. It will basically send out electrical signals on its own to override your heart signal, and then it restores normal heart rhythm. So how do we detect the electrical signal from the heart? Does anybody know what medical procedure we can do to check the heart signal?
ECG.
Echocardiogram.
That's right, ECG. So ECG is electrocardiogram here. Okay? It's a non-invasive way. That means like they don't have to put anything in you. It's just these little stickers that they put on your body, and you know, in the like a few stickers closer to the foot, and it measures the electrical signal generated by your heart. And this is a typical ECG graph. Okay? So there are a couple of things that I want you to pay attention to on this graph. Right? So there's a little peak here, and then there's a sharp spike there. I want to show you what those corresponds to. What event does those points corresponds to? So here, first, the pacemaker, as in like your pacemaker. SA node is the natural pacemaker, right? The one that you implant, that's called artificial pacemaker. Pacemaker means it sets the pace of your heart rate, right? Basically, that's what it means. So when the SA node sends the signal across the atria, that causes your atria to contract. We talked about that already, but it's not a very strong squeeze. Right? You don't need to squeeze very hard because the ventricles are just below. Gravity does most of the work for you, right? So that squeezing is basically this part of the graph. So on the ECG, the first bump corresponds to atrial contraction. You need to know this, okay? Corresponds to atrial contraction. And then there's a little bit of like delay. That's like the signal is being passed down to the ventricles, to the apex, to the pointy end. And so it's going to go through the AV bundle, It's going to get there. It's going to take some time. Not too long, but it does take some time. So nothing really happens there. And then once it gets to the ventricles, then that's when we have this big spike. Okay? This big spike is ventricular contraction. Okay? You can see it's much higher, the peak. That means the contraction is much stronger compared to the atria, which again should totally make sense to you because the ventricle needs to squeeze the blood all the way throughout the body, right? On the left side anyway. And on the right side, you have to squeeze to the lungs. Sure, the lungs are nearby, but still you need to generate enough force to send the blood there, right? So compared to the atria, the ventricle needs to squeeze much harder, which is why there is a bigger peak. Okay? And then don't worry about the rest of it. The rest, like, I'm not going to explain it, okay? But basically, the heart needs to reset itself before it can go through another round of excitation. That's what it's called, okay? Like, before it can respond to electrical signal again, it kind of needs to reset itself. So there you have it, the ECG. Now, here are a bunch of ECG that looks abnormal, okay? I probably should put in like the normal one for you, okay? But then like, I mean, if you look at this, you don't have to be like a, medical expert to kind of know that this is not right, right? Like the peaks are too far apart compared to like one cycle of these things, right? So this corresponds to someone who is having slow heart rate at 46 beats per minute. That's really slow, okay? So that's called bradycardia. You don't have to know any of these names, okay? I'm not going to test you on it. I'm just trying to show you, okay? Don't have to know. Don't have to know. Okay? So bradycardia, too slow. This is tachycardia, too fast. Brady anything is too slow. You can have too slow of breathing, too slow of the heart. You use the prefix brady. Too fast is tachy, okay? Tachycardia. And then over here, this is like the worst kind, okay? This is ventricular fibrillation. Your ventricle's not even squeezing. They are like going into spasm and it's not delivering blood. Person's gonna die soon, right? So to save someone who is having a ventricular fibrillation, you need to have the defibrillator. On TV, sometimes you see the people with the thing, and then the doctors say clear, and then they shock the person, right? So nowadays they have these AED. Okay? It's automated electric defibrillator. Right? You just put the pads on and you press the button, and then the machine does the rest for you. Okay? You can find them at schools, shopping malls. They're kind of everywhere because it could be a life-saving tool, right? And they are again fairly easy to use. So that's ECG and electrical signals. Anything you want to ask before we move on to the next difficult thing? There's always the next difficult thing. I'm so sorry. Any question?
I have a question, Professor.
Mm-hmm.
In the electrodiagrams ECG,
Mm-hmm.
this is for normal person. The
Oh,
one is
these are all abnormal. These are all
abnormal. Okay,
abnormal. Yeah.
okay.
So the normal one would be like closer. Yeah, I probably should include like a normal one for you as reference. But again, right, don't worry about. I'm never gonna show you a picture of these and ask you like what's wrong with that person. I'm just showing you these as examples. Okay, you don't have to know. Okay, let's see if you remember this one. We did some calculation last time. Let's see if you remember. All right, most of you got it right. Cardiac output is milliliters per minute. Okay. Cardiac output equals to heart rate multiplied by stroke volume. Heart rate is beats per minute. Stroke volume is milliliters per beat. If you work it out, then cardiac output is milliliters per minute. Okay, next question. Good. This one, again, should be pretty self-explanatory. If you're exercising, your heart rate goes up. And since cardiac output equals to heart rate times stroke volume, if heart rate goes up, cardiac output goes up. The answer is A. Okay, that's it. That's it. Let's move on to blood vessels now. Blood vessels. That's all the blood vessels in the body, okay? Like if you remove everything else, that's blood vessels. So functions of blood vessels. Some of it's pretty self-explanatory. For example, the blood vessels are used for transporting blood and whatever is inside the blood. Okay, we're gonna learn all the contents of blood next week, but it's used for transport because we have a closed circulatory system, right? All the blood is gonna stay inside the blood vessel, so that's pretty obvious, I would say. The blood vessel also helps carry out exchange of gases in the pulmonary capillaries and exchange of gases plus nutrients for waste at the systemic capillary. Pulmonary capillary means it is at the lungs. Okay, and at the lungs, what you will be doing is you will be adding O2 and removing CO2 from blood, right? Systemic means bodily. So when you are talking about capillaries in the body, right, like at tissue, body tissues, what we're doing is we are removing O2 from blood and adding CO2 to blood so that you can carry the waste, CO2, back to the lungs where you breathe it out. Okay? So these kind of exchange only happens at the capillaries. There are no exchange between the arteries and the surrounding or the veins and the surrounding. You can only exchange things with the surrounding at the capillaries. Okay, capillaries are like the smallest blood vessels, right? We wrote it down last class. You know, heart, arteries, arterioles, capillaries, venules, veins, back to the heart. We wrote it down last time. Okay, next, blood vessels can help regulate blood pressure, and more on that later, but they can get bigger or smaller, which in turns change the blood pressure. And blood vessels can also direct blood flow to where they are most needed in the body. Right? Your body doesn't need 100% blood delivery at all times. Like right now, I assume you are sitting somewhere, okay, watching me talk. So there is no need to send too much blood to your legs because you're not running around, right? Okay, so your blood vessels can help divert the blood to other places, maybe to your digestive system. If you had lunch before you come to class, and maybe you're digesting your food now, so we need more blood going to the digestive tract. Okay? When you're running, when you try to catch a bus, when you're trying to, you know, run away from, I don't know, like a dog or something, then you need more blood to your muscles, right? And not so much blood to, say, your digestive system. So the blood vessels can help you kind of redirect the blood flow as necessary. So I want to show you this picture. A couple things are important here. Let me just change the color so you can see better. Over here is the blood. The blood is going into the artery, okay, and then it's gonna go into the capillary here before returning to the veins. Now in the capillaries, you have these sphincters. Okay, sphincters. A sphincter is like it's kind of like a valve. Okay, you can open them, you can close them. So let's say whatever this part of the body is. Let's say this part of the body needs a lot of blood. So you would open all these sphincters, and the blood would come in, and then it would completely fill up the capillary like this. Like you don't have to draw this, right? Obviously, just watch what I'm doing and try to understand. Okay, so you're gonna fill it up. You're gonna give it like 100% supply of blood. But let's say you don't need this part. You don't need to use this part at the moment. Then you can close off. You can close off some of these valves, these sphincters. Okay, so let's close it off here. Close it off here. It's not like we are not giving blood to this section, but now instead of filling up the whole network, we're just gonna provide the minimum amount required to keep it alive. Do you understand? It's like turning on the tab, right? Like you don't always turn the tap on to max, right? Like if you're just washing some like a cup or something, you just turn on the tab a little bit and then wash it. If you turn it up to max, it's gonna go all over the place, right? It's unnecessary, right? So that's what these sphincters are. It's like flow valves. Okay, how much blood should we supply to this location? Does it need a lot? Does it need a little bit? Right? So the blood vessels help you regulate that. You don't even need to think about it, right? Your body does it for you. Okay.
Vincent, so these precapillary sphincters are like blood vessels too.
They are part of the blood vessel. That's right.
Oh, okay.
Yeah. So let's take a closer look at this picture right here, right? We have the artery right here, and we have the veins here. Okay, they are not like really those kind of colors. Okay, when we prepare microscope slides and we look at different features, we can stain them using different techniques. Okay, so we purposely make, I mean, not we, but like the person who made this thing up, they purposely use a very vibrant red to highlight the artery and blue for the veins. Okay, so it's like you have the blood vessel, and then they cut it. That's why it looks like circle, right? Can you give me some difference that you can observe between the artery and the veins?
Why the size is different, Professor?
Yeah,
For
the size
example,
is different.
Yeah.
Yeah, that's right, right? So like artery, I would say smaller diameter, right? And then over here we have like bigger diameter. Okay, so that's a pretty obvious difference. Is there another difference that's a little bit more subtle that you notice?
The artery is thicker than the vein.
Mm-hmm. That's right. So here is, even though they're smaller diameter, it's gonna be thicker walls. Okay? Right here, you can see much thicker, and this is thinner, right? Thinner wall. And one of the reasons is because the blood pressure, as you will learn, in artery is much greater than that of the vein. And if the wall is too thin, you're gonna burst the blood vessel, and that is not a good thing. All right, we're gonna take a very long note next. Okay, it's called capillary exchange. Hopefully you have some colors of, I would say like at least 3 colors would be nice. Yeah, don't do it in black and white. It wouldn't really make sense. So here we go. So I would suggest like starting a new page, and probably better if you do it sideways. Okay, so we're gonna write some notes here, and then we're gonna draw, and then we're gonna write some more notes. Okay. Anyway, I'm just telling you so that you can plan your space ahead of time. So here we go. Capillary exchange. First, like, what does it mean? Like, what is capillary exchange? Capillary exchange is basically movement of substances, I guess. And fluid in and out of capillary. Okay, so what goes in, and what goes out of the capillary. That's basically what capillary exchange means. Okay, we're gonna look at 3 factors that affect capillary exchange. There are 3 things that will affect what goes in and what comes out. Now, don't think of the capillary as like a water hose. Right? People think of blood vessels as water hose, right? And you know that's okay, in a way, because you know you're probably thinking that the blood is not gonna leak out of the blood vessel, right? Just like the water is not supposed to leak out of the water hose, right? Yeah. So that's true for the arteries and the veins. But capillaries are different. Capillaries, they are by design leaky, okay? But it's not blood that's leaking out, as you will see. Okay? When I say they're leaky, it just means that it's not like a wall, right? Like a wall behind me, right? Like nothing goes through a wall. But the capillary is leaky in a way that it allows some things to pass through. Okay? So 3 factors that affects capillary exchange. I'll list them here first, and then I'll explain what each one means. The first one is easy. Okay, first one is diffusion. You learned diffusion last semester, okay, but I'll explain again. And the second one is called it's blood pressure. Blood pressure. And the third one is probably new to you. It's called, excuse me, it's called osmotic pressure. I'll explain, okay? I just want to write them out for you first. There you go. So let's talk about diffusion. What's the definition of diffusion? Anybody want to give me their best definition for diffusion? What does it mean? What does it describe?
Movement of molecules from higher concentration to lower concentration.
Yeah, exactly right. Movement of substances, molecules, whatever you want to say, particles, substances, from an area of high concentration to an area of low concentration. And it just happens on its own. No energy is required. It's spontaneous. Okay. So let's say we have a capillary here. I know it's supposed to be like a net, okay, but it's so too hard to draw. So I'm just gonna draw a tube here, okay? This is capillary. Here, capillary. And then this is like the surrounding tissue. Okay, so we'll say systemic tissue. like, of course, this can happen in the lung as well, but I think let's just use systemic tissue. Like it could be the liver, it could be the muscle, whatever, it doesn't matter. Okay. Now let's say the blood is coming in this way. This is blood flow. So the left side of the capillary is connected to an arterial, right? So we call this the arterial end. And then the other side is connected to veins, which we'll call the venous end. If this is a capillary that's found in the body tissue, then it's gonna be, do you think is going to be oxygen rich or oxygen poor blood? This blood is coming in to the tissue. Oxygen rich or oxygen poor?
Rich.
Because we have to deliver the oxygen to the body, right? So, you know, if you have colors, then I'm gonna put some O2 here. O2, O2, O2 is it's rich in O2. Yeah. And then the systemic tissue, it will have some O2 as well, but not as much because you have been using it, right? And this one will have a little bit of carbon dioxide, but not a lot. But then you're gonna have a lot of carbon dioxide in the systemic tissue because again, you're doing cellular respiration, right? Which takes glucose and oxygen, producing carbon dioxide and water. Okay, so when this happens, what will end up occurring is the oxygen is gonna diffuse out from high concentration to low concentration. And then the carbon dioxide is gonna diffuse in. Okay, so afterwards, the blood is gonna have more carbon dioxide. And then less oxygen, right? And it's going to be the other way around for the tissue, but I'm not going to draw it so that's not going to be too confusing, okay? So essentially what happened is that you have red blood, so to speak. All bloods are red, of course. And then they become blue, right? Again, not literally, but like, you know, figuratively, I guess. Okay, blue blood is oxygen poor, right? Yeah, and then this is gonna basically go back to lungs, right? Back to the heart and back to the lungs. So that's diffusion. It's very simple, right? It doesn't have to be just oxygen and carbon dioxide. It could be nutrients. It could be glucose. You might have glucose-rich blood here. And then, you know, the glucose will diffuse out into the tissue where you then can use it for cellular respiration. So that's the easy one when it comes to factors that affect capillary exchange.
Vincent,
Mhm.
yeah, you said systemic tissue also means bodily tissue, right?
Yeah. That's exactly what
okay.
it means. Yeah.
Yeah, so does that mean the capillaries in the body or the one in the heart, which one are we, like talking about here? Or is it in general?
Say that one more time. I didn't quite catch
Okay,
the
I
last
mean this capillary illustration,
part.
is it for the one like taking blood to the whole body or?
Oh yeah. So it's taking the blood to the body, but like which part of the body I didn't specify,
Which
right?
one? Yes.
Yeah, it could be like the liver, it could be like the muscle, it could be like the legs,
Okay.
right?
So that means we have different capillaries for each part of the body.
Yes. Now remember last time, I think it was you who asked me this question, but let me just go back here. Like this diagram, right? Remember? Right?
Yes.
Yeah. So like all you have capillary that's designated for the kidney. You have one designated for like so on and so forth. Right?
Okay.
So all these are like systemic, right? Okay. Any other question with the diffusion stuff? I hope it's, you know, Reasonably straightforward, huh? Now we're gonna talk about blood pressure next, number 2. And I need you to draw a new diagram, okay? New diagram, the bigger, the better. Blood pressure. In a bit, okay? Let's take some note first. What is blood pressure? Okay, blood pressure is the force of blood pushing against the vessel wall. Okay, if you think about like a balloon, okay, think about balloon. I mean, you can feel the pressure in the balloon if it's inflated. That's basically like the gas pushing on the balloon, right? Okay, like if you fill it up more, then you feel more pressure pushing, right? So in this sense, blood pressure is related to blood volume. The more blood you have, the higher your blood pressure. Yesterday, someone said blood is about 5 to 6 liters, right? More blood you have, higher the blood pressure. Right? But blood pressure is the force of blood pushing against the vessel wall. Now, I want you to go through this mental exercise with me. Okay? You can draw it if you want, or you can just listen and try to visualize what I'm saying. Okay? So here we have a tap, okay, water tap, and I'm gonna hook up a water hose to it, like one of those really long hose that you might have, you know, to water your lawn or to wash your garbage bin or something, right? Really long hose, okay? Now in this water hose, this is a water hose. In this hose, we are going to poke some holes into it. Sounds like a tongue twister. We're gonna poke some holes. You're gonna take something sharp and you're gonna poke some holes into it. Poke some holes. Okay? Just like visualize this. Now, we're gonna turn on the tap all the way to maximum, as high as possible. Think about what's gonna happen next. Okay? Visualize, imagine what's gonna happen next. Now, common sense tells you water is going to be spraying all over, yes? But is it going to be different amount of water coming out of these holes, or do you think it's about the same?
It's different.
It's different,
And the
right?
first, it's high pressure, and then
Exactly.
when it's come down, it's reduce the pressure.
Very good. Okay, so like at the beginning, when it's closer to the hole, it's like spraying all over. Okay, 5, 6 feet high in the air. Okay, I'm not going to draw all of it. Okay, but maybe in the middle, it's like, you know, it's still spraying quite a bit. But if it's really long hose, eventually further down, it's just gonna be like, you know, coming out a little bit, right? Does this make sense? Now, what does this have to do with what we're talking about? Well, in this analogy, this is like the heart. Okay, and then the heart is connected to a bunch of blood vessels, right? Starting with the aorta, and then you're gonna have some artery, you're gonna have some arterial, and then capillary, venule, and then veins, right? Again, we wrote this down last class, which I just did it in a circle last time because eventually you go back to the heart, right? Okay. So when you turn on the tab, it's like the heart pushing the blood forward, right? And then just like the analogy I'm trying to tell you, the blood pressure is gonna be high here, higher BP. And then eventually you're gonna have lower BP, right? Because you're further away from the source of the pressure. So as blood moves further and further away from the heart, the blood pressure is going to drop. Right?
Is that how the blood pressure is measured?
Not entirely. But yeah, I think we're gonna get to that later on. Okay, so keep that in mind. As blood moves away from the heart, the BP, the blood pressure drops. Okay. And then what is the effect of blood pressure on capillary exchange? Well, just like the water hose thing, you squirting water all over the place. Blood pressure pushes fluid out of the capillary. Okay. Now, it's not blood that you are pushing out. Like that would be like internal bleeding. Okay. We're just pushing out some like the fluid portion of the blood, a little bit of it. Okay. Blood is made up of mostly water, right? You will learn that next week. Okay, but it's mostly water with some cells and some nutrients and some proteins in it. This blood pressure will be pushing out some fluid. Okay, some fluid. So let's draw that. Okay, so I need you to draw a new diagram, a big one if you can, because we're gonna add a lot of details to it. Okay, so we have this capillary again. Don't use the other one. Okay, you need a new one for this. You're gonna have the arterial side. You're gonna have your venous side. And to remind you that the capillary is leaky, I'm gonna put some gaps here. Okay. It's not so leaky that you're gonna be bleeding out from them, although that sometimes can happen. You know, sometimes people with high blood pressure, the capillary in the eyes might burst, and then the eye becomes red, right? Because the blood is leaking out, basically. So that can happen, but that's not what we're talking about here. Okay, it's normal for the capillary to have a little bit of a gap in the walls. Okay, so I'm gonna use red arrow to represent the blood pressure. So in the arterial side, the blood pressure is gonna be pushing fluids out. Okay, it's gonna be pushing the fluid out. There we go. Okay, gonna be pushing it out, pushing it out. But as we move further and further away towards the other side, right, the blood pressure is gonna gradually drop, gradually drop. Okay, so the blood pressure here, I'm just gonna give some numbers, approximately 30 mmHg. mmHg is millimeter mercury. That's a unit for measuring blood pressure. And the blood pressure here is approximately 15 mmHg. The numbers are not like, I mean, don't need to memorize them, okay? I just need to tell you the numbers so I can explain something else to you in a bit. mmHg. mm is millimeter, right? And Hg as in like from the periodic table, mercury. Okay. Any question before we move on to the third factor? Really? Everybody understood that? That's great. Okay, we're going to come back to this picture in a bit, but now let's talk about the third factor. Osmotic pressure. Osmotic pressure is generated, or we can say caused by a difference in solute concentration. You might remember this discussion last semester. At least I hope you do. Let's say there is a membrane here, okay? And then I'm just gonna fill it up with some water. There we go. Semi-permeable membrane. Oh, hold on a second. Sorry. I forgot. To tell you to leave some space. Okay, we need some space there. If you didn't, I guess that's okay. Okay, but but if it's still possible to change it, leave a little bit of space. All right, I'm gonna put some solute in here. Okay, some solute. These are very big. They cannot cross the membrane. Huh? So we have higher concentration on the right side and lower concentration on the left side. All right, let's see if you remember this. Which way is the water going to go? Which way is the water going to move? Is it this way or this way? Which way will it move? The water, okay? Not the purple balls. The water. Anyone else? Okay, well, the correct answer is water moves towards the more concentrated side. To try to dilute it, right? To balance it out. Right? So in other words, it's going to suck the water towards that side. Okay? So as water moves to the right side, it will dilute the right side and it will concentrate the left side. This will continue until the concentration balances out. Okay. And this ability to suck water towards one location creates the osmotic pressure. Okay. So the concentration of blood, okay, so with dissolved ions, right, and cell and proteins, and cellular and cells, blood cells is higher than the concentration of the surrounding tissue. So does this draw fluid into the vessels or does it push it out? Is it going to cause fluid to move into the capillary or move out of the capillary? Do you guys understand? Let me
Move in.
move in. Yeah, move into. That's right. Move into the capillary. Okay. Let me show you on this. Okay. I'll let you copy in a second. What I'm saying is, if you consider all the stuff that's in the blood, you know, there's a lot of ions, right? Sodium, chloride, potassium, calcium, magnesium, all these stuff. And then there's like cells in there as well. There are a lot of proteins in it, right? So all these are like components of the blood. That's much higher compared to the surrounding. And so as a result, you're gonna be drawing fluid into the capillary. Okay? That's what I mean. Okay, we'll finish drawing the arrows in a bit, but copy what you need to first. And, you know, ask questions. I keep on reminding you to ask questions. Okay, back to here. Now, the concentration of the solute doesn't actually change substantially from one side to the other. Okay. And the solutes are too big to leave on their own anyways. Okay. Like things like gas, they could exchange, but you know, the cells are too big to leave, right? So for the most part, the OP, it's about 20 mmHg here, and it's gonna stay the same throughout. Okay, so I'm just gonna draw the same arrow. Okay, same arrow all the way across. Not gonna be different. There you go. Okay. Maybe let me make this one a little bit shorter so that it's more obvious when I try to explain to you what's going on. There you go. So the OP here, the osmotic pressure, OP is approximately still 20 mmHg. Not much change from one side to the other. Okay, so you can think of OP and BP, blood pressure and osmotic pressure. You can think of them as opposing forces. Blood pressure is always pushing things out, and osmotic pressure is always bringing things in. Okay, so they're counteracting each other. So depending on which pressure is stronger, you will either be losing fluid or you'll be gaining fluid. So let's take a look at this end here in the arterial side. In the arterial side, the blood pressure is greater than osmotic pressure. Therefore, do you end up losing fluid or gaining fluid?
Losing.
That's right, you end up losing fluid. So you have a net loss, net fluid loss. Okay? And that's what's going to be happening. You're going to have fluid coming out. Not blood, just fluid, okay? over here on the venous side, the blood pressure is going to be less than the osmotic pressure. And therefore, you will have net fluid gain, right? You're going to be bringing the fluid back in. Is that okay? Now, for those of you who have a very sharp eye, you might notice something of a problem here. If you do the calculations, 30 minus 20 is 10. Okay, so the pressure difference here is 10 mmHg. On the other side, the pressure difference here is 5 mmHg. So the difference is greater on the arterial side, which then tells you what? There's a difference of 10 unit of pressure here and only 5 unit of pressure difference here. What does that tell you about this process? No? Well, what it tells you is that you actually end up losing more fluid than you can bring back, right? Because the pressure to bring them in is actually less compared to the pressure that's pushing it out, right? So you end up losing more fluid on the arterial side than you are able to bring it back. Okay, so what happens to those excess fluid? Well, you will have to wait until lecture 2. Okay, we'll come back to this concept later. If I cannot explain that right now. Like I have to teach you a bunch of other stuff because before I can explain it to you why. Okay? But just keep that in mind. You don't actually bring back everything you've lost. Okay, and that's it. That's capillary exchange. Let's do 2 poll questions and see if you learned anything. There we go. Okay, please participate if you can. If you don't see the poll, you can type the answer in the chat box as well. Okay. It's important to participate. I only have 4 answers. Where is everybody? The answer is right in the notes that we just took. All right. So this is asking what is true in the arterial side. So if you take a closer look at the notes in the arterial side, which is right here on the left, the BP is greater than the OP. So you end up losing fluid, right? So the answer is BP greater than OP, net pressure out, as in you are pushing fluid out. So the correct answer is A. Here's another one. Okay, osmotic pressure is the green arrows. Think about the note. Do they change from one end to the other? Okay, look at the osmotic pressure, right? It's 20 mmHg on the left, and it's also 20 mmHg on the right. So it's the same. Right? Why is it the same? Well, because the concentration doesn't really change from one side to the other. Okay. As a result of that, the osmotic pressure is the same throughout. The answer is C. Okay. Any questions before we take a break? I feel like this this is not 100%. Some of you doesn't really fully get this. Anything you want to ask before we take the break? Okay. Well, if you think of anything, you let me know. Let's take a 5-minute break. We'll come back at 1:47. See you soon. Okay, let's continue. During the break, did you guys think of anything you want to ask before we move on? We're done with this concept, okay? We're moving on to something else. Any last minute question? All right then. Next, we're going to talk about venous return. Venous return is the term that we use to talk about bringing blood back to the heart. Okay, bringing blood back to the heart, and that can be challenging for the lower part of the body, because the heart is up here, so everything that's below that point has to go upward against gravity in order to go back to the heart, right? And to add to that problem is low pressure. We already learned as blood gets further and further away from the heart, the pressure drops continuously, right? By the time you get to the veins, the pressure is really low. Right? So with low pressure, it's hard to move things up against gravity. Like, think about it. If you have like a juice box, okay, the liquid is not going to automatically come out of the straw, right? But if you apply a bit of pressure, if you squeeze it, then all of a sudden it squirts everywhere, right? So with high pressure, it's okay, you can move things upward against gravity. But with low pressure, as is the case with the veins, it's harder to move things against gravity. So to overcome this, there are two mechanisms, two ways to help fix these problems. So number one, to help with the pressure situation, a lot of times the veins are surrounded by muscle. Right? Certainly that's the case for the leg muscle. So when you are walking around, when you're contracting, squeezing those leg muscles, those contractions from the muscle actually help squeeze the blood in the veins and squirt them upward. Okay, so that's called like a skeletal pump. And that's not strong enough to get the blood all the way back to the heart, but it's going to help it go up a little bit. Okay, and when you relax the leg or relax the muscle, then what happens is the blood will drop back down, but not all the way down, because in veins we have valves. So when the muscle contracts, it will squeeze through the valves, it will go up. But as soon as they relax, the blood will come down and the valves will close and catch the blood. So next time when your muscle contract again, you don't have to start all the way from the bottom. You can just continue moving up from wherever you left off from the previous contraction. Okay, so a little bit by a little bit, eventually the blood's going to move all the way back to the heart. So that's called venous return. Now, for some people, the valves might be not working, okay, and that could lead to what's called varicose vein. In varicose vein, you can see the veins are bulging out because the blood is not emptying in the veins. They are pooling in the veins. The valves are leaky. They're not able to, trap the falling blood, so to speak, and so they stay in the vein. Right? To fix this problem, people will have to wear these elastic stocking or elastic stretchy socks. So that extra bit of elasticity will help basically serve as a pump, right, to help empty these veins. Okay. Let's talk more about blood pressure. We already know blood pressure is the pressure exerted by blood against the wall of the vessels. Okay? The more blood you have, the greater the force, the higher the blood pressure. We also know that the blood pressure is going to drop as it moves further and further away from the heart. And really, when the ventricle contracts, that's what's creating that blood pressure initially. So there are two pressure that we are interested in when we measure blood pressure. There's the systolic pressure and then the diastolic pressure. The systolic pressure is the pressure that we measure when the ventricle contracts. Okay? Pressure measured when ventricles contract. And that is usually the higher number, not usually, that is the higher number, okay? That is the higher number. And the diastolic pressure is when the ventricles relax, and that would be the lower number. And we measure blood pressure in the arm, right? Typically, it's also possible to measure it like in the leg in some cases, but typically they do it in the arm. So there are many things that can affect the blood pressure. Here are just a few of them. Total blood volume. As I told you already, more blood translates to higher pressure. Less blood, less pressure. One of the reasons why dehydration is dangerous is because it causes your blood pressure to drop dangerously. Not enough blood pressure, your blood's not going to go through the body quick enough. And then, it might not go to your brain, you might faint. Stroke volume, that's basically how strong your heart contracts. Okay. The stronger it contracts, the higher the blood pressure. Vessel diameter. Okay, so we can have vasodilation. Vasodilation means the blood vessel gets bigger. So that equals to larger diameter, which equals to higher blood pressure. Oh, sorry, which equals to lower blood pressure. More room, right? Less pressure. Opposite of vasodilation is vasoconstriction. You are squeezing the blood vessel. So smaller diameter, which translates to higher blood pressure. Okay. And as I said, we measure blood pressure in the unit of millimeter mercury. And we have the systolic value, which usually they say is 120, and then 80 is the diastolic pressure. Okay, but these numbers are relative. Okay, it really depends on the person. Right? Someone who is younger, they might have better numbers. So that's blood pressure. Now I'm going to show you 3 graphs. They have quite a bit of information in it. Hopefully it's not too confusing. The first graph here, this is plotting blood pressure versus distance from left ventricle. blood pressure and distance from left ventricle. We already explained this earlier, right? The pressure is highest in the aorta, and then it drops. But this graph shows you additional information. Okay? You can see here at the beginning, the blood pressure fluctuates. Right? We have the higher systolic pressure and the lower diastolic pressure. But that fluctuation kind of diminishes as we move further away from the heart. And in fact, by the time you get to the capillary, the blood pressure is the same, regardless of whether the heart is contracting or relaxing. Okay. By this point, you are so far away from the heart that you no longer feel the pressure difference from the contraction or the relaxation. Okay, so it's only at the beginning that you have two numbers. Right? By the time you are far away enough, you only have a single number. So that is the blood pressure. Next, we have velocity change. Velocity is basically the speed of blood flow. Speed of blood flow. Okay. So it depends on two things. Depends on, number one, blood pressure. Okay. And number two, vessel diameter. Okay. So high blood pressure equals faster. Again, that's not surprising. Okay. Think of the juice box again. You turn the juice, take the straw out, turn the juice box upside down, start leaking, right? If you want it to leak faster, you squeeze in it, right? And then it shoots out. Squeezing it increases the pressure, the flow goes faster. What about vessel diameter? This one people usually have a hard time. If it's a big vessel, do you think the speed is faster or slower? Don't think about the blood pressure at all. Just think about like the size. If it's bigger, do you expect the blood to go faster or slower?
Faster.
Faster, that's right. Think about driving with 6 lanes as opposed to 2 lanes, right? 6 lanes gonna have faster traffic, right? So bigger equals faster. Right? So the velocity is gonna fluctuate at the beginning, not surprising, because it depends on blood pressure. And you can see in the previous graph that the fluctuation occurs at the beginning. So as blood pressure fluctuates, so does the speed. Okay. And then it drops significantly at the capillaries because they are really small and the pressure is not that big. But then it picks up again. It speeds up again when we get to the venules and the veins. Okay, because the diameter gets bigger and bigger, right? So it picks up the speed. So velocity is going to be the slowest in the capillary. Okay, slowest speed. Okay. Slow enough for gas exchange to happen, right? Again, it should make sense. If the blood is going by like super fast, how are you gonna have enough time to get the oxygen out and put the carbon dioxide in, right? You need to slow it down, not come to a stop, but slow enough to allow the exchange to take place. Now there is a third graph here, which is this one. And this one that goes up and comes back down, okay? This is total cross-sectional area of the vessels. What that means is, like, if you lay out all the vessels, how much area is it gonna cover? Right? So aorta is really, really big, but you only have one of them, right? So that's why it doesn't really cover too much area. Capillaries, they are small individually, but you have a lot of them, okay? Virtually your whole body is covered by capillaries. Okay? No cell is 2 cells away from the capillaries. It's basically everywhere. Like when you have a paper cut, you basically cut the capillaries. Okay, so like everywhere is capillary. So you have a lot of them, and collectively, if you add up all the area that they cover, it's a lot. All right, so there you go, 3 graphs. Hypertension. Someone has high blood pressure if they are not able to regulate blood pressure. We have different ways to regulate blood pressure in the body. Your kidney is going to help with that. You have hormones that's going to help bring it down. And all these things is going to try to, you know, keep it at 120/80. Right? But if all these things fail, then your blood pressure is going to go up. Right? If the resting blood pressure is above 140/90, then we say if this person has chronic high blood pressure. So high blood pressure for a long time is not good for the heart, is not good for the kidney, increases the risk of heart diseases as well as kidney failure, extra strain on the heart, obviously. As for the kidneys have very delicate capillaries, as you will learn later on. And if you put too much pressure into it, you're just going to destroy them, essentially. So what contributes to high blood pressure? Some of it is hereditary. You're just born with bad genes, and there's not much choice in that. But diet is a significant contribution as well. Okay, what you eat, the level of physical activity you have, right? All those things can contribute to high blood pressure. If you suffer from high blood pressure, what kind of food should you avoid?
Party food?
Salty food.
Salty food, that's right. Yeah. So why is salty food not ideal when you have high blood pressure? Well, think of the capillary exchange thing that we did before the break, right? If you have a lot of salty food over time, where does all those salt go? Salt's going to be in the blood, right? Right? So basically, you are going to be having extra dots, extra solutes in the blood. And if that's the case, then you have higher osmotic pressure, right? Higher osmotic pressure. And osmotic pressure basically draws fluid into your blood vessels. That will increase your blood volume, which will then increase your blood pressure further, right? Does anybody know what kind of drugs people take when they have high blood pressure? I don't mean the name of the drug necessarily, but like, the kind of medication that they take. What does that do? What does that help them do? Anybody know by any chance? Has anybody heard of the water pill? Water pill. The proper name is diuretic. Anyone? No? Okay. Water pill is again medication for people with high blood pressure. And when you take it, it makes you pee more. How does peeing more help with high blood pressure? What's the main thing that's in the pee? What's the main thing that's in the urine?
Sodium, like salt?
I wouldn't say so. Like it's mostly. Anyone else want to try?
Like,
What's that? Sorry.
no, I mean water and salt, like together.
Yes, mostly water. Exactly, right? Mostly water. So when you pee a lot, you're basically reducing the water content in your body. Okay, and so your blood volume goes down, and blood pressure goes down. Right? All right, let's do this. Go ahead. Anyone else? Usually I get 8 answers. All right. So the correct answer is veins. Okay, blood pressure is the lowest in veins. Look at this graph. Okay, blood pressure is going to drop, and in veins is going to be the lowest. How about this one? Okay, good. The speed is the slowest in the capillaries, right? Remember I said you have to slow down enough for the gas exchange to take place. Another one. So remember, the speed is dependent on two things, pressure and the size. The higher the pressure, the faster. The bigger the diameter, the faster. So if you can have a vessel with high blood pressure and large diameter, that's going to be the fastest. Okay, and that is the aorta. In the aorta, it is the fastest, right? Look at here, right? At the beginning, right? It never really quite reached that point again. So aorta is really big. I showed you a picture of it, right? And the pressure is really high. Speed is going to be super quick. All right, let's do this one together. High blood pressure. Is it artery or vein? Well, artery is closer to the heart. It's going to have higher pressure. Because of the high pressure, it needs to have thicker walls to withstand them. Otherwise, it will burst. Larger lumen. Lumen is the inside, okay? Like any structure that's hollow, the inside of the hollow organ is called the lumen. Like if this is the blood vessel, this is the lumen, okay? The lumen, the inside. Your stomach lumen is where you hold the food, okay? Lumen is just, again, the inside of a hollow structure. So if it's larger diameter, it's the vein. I showed you that picture earlier, right? With the vein besides the artery. You can see it was much bigger. Which one has valves? Does anybody remember?
Vein.
The vein has valves, right? Because of venous return, it needs to trap the blood so it doesn't fall the way down, right? Okay. For like we're talking about general artery and veins. Okay. Some people get confused. They're like, hey, but the aorta has a valve and the pulmonary artery has a valve. Those are like, you know, for all intents and purposes, those are like valves related to the heart. These are like veins that is like elsewhere in the body, okay? Higher velocity, I hope you remember it's artery and carries blood away from the heart. That is also artery. Okay. So each lecture, I try to teach you the anatomy, like what is it called? So we labeled the heart last time. I teach you the physiology, like how it works. Right? So we talk about the pumping, the squeezing, the conduction system, the capillary exchange, and then I also will talk about I'll talk a little bit about diseases, okay, the pathology. So we're going to talk about cardiovascular disease. Cardiovascular disease is actually the #1 cause of death in North America for many, many years. And a lot of people are actually affected by it. And you cannot talk about cardiovascular diseases without talking about cholesterol. So there are two kinds of cholesterol. Cholesterol, as you know, it's a type of lipid. And because it's lipid, it's not soluble in water. Your blood is mostly water, right? So the cholesterol gets processed in the liver and it gets combined with these lipid carriers, okay, that are made up of protein. So it's gonna become LDL or low density lipoproteins. This is the bad cholesterol. Sorry, bad cholesterol. You don't want this. And then the HDL, that's the good one. Okay, it's good, but then you don't want to have too much of it either. Okay, so the bad cholesterol tend to build up in the endothelial lining. Endothelial is just like the inner lining of the arteries, and whereas the other one, you know, we use it for various things, including forming the biles. If you don't know what bile is, it's in the gallbladder. It's greenish yellow, helps you digest fat. So we have what's called atherosclerosis. Atherosclerosis is the hardening of artery caused by a buildup of lipids in the arterial walls. Okay, so this is the hardening of artery. And then the blood clot is gonna form on top of this fatty buildup. Right? So it's like this. This is a blood vessel, and let's say you have a little bit of fat buildup here. Right? So this is a fatty substance, fatty buildup. It could be cholesterol. It could be, you know, some of the like the saturated fat that we talked about, right? And I told you before, blood does not like non-smooth surfaces. As soon as there is a little bit of fatty buildup here, then you're gonna have things that sticks to it. The blood, right? The platelet's gonna stick to it, and then it's gonna become like a snowball effect. It gets bigger and bigger and bigger and bigger. Okay, so this is a blood clot, and before you know it, your blood vessel is gonna be narrow. So the blood is now gonna be like squeezing through it, right? And that's basically what atherosclerosis is. This part of the blood vessel becomes weaker and it's hardening. When it hardens, it loses elasticity. It's not able to withstand pressure as good. And sometimes, if this is really severe, it's possible that the blood pressure is too high. It will cause it to rupture. Depending on where it is, it could be deadly. You might have heard of people who die from an aneurysm. An aneurysm, aneurysm is basically one of these things in the head, and the vessel is really weak, and any kind of external trauma, like if you bump your head, you fall down, you bump your head, that trauma is strong enough to break the vessel, and then the person have bleeding in the brain, and they could die. Okay, like almost immediately, right? So that's dangerous stuff, right? But sometimes this just happens, you know, elsewhere in the body, and it could contribute to heart diseases. So here is a healthy blood vessel. Okay, they cut it open, and then you can see the lumen, the inside, is unobstructed. But for someone with atherosclerosis, you have the atherosclerotic plaque, which is the fatty substance. That's just the start of it, right? Like what's really blocking your vessel is it's not entirely the plaque, but the blood clot that you form afterwards. Right? And you can see like it's really, really narrow. the remaining space, this white space here, that's all there is left for the blood to squeeze through. And you can understand why this could be a problem, right? So last class at the beginning, I told you the heart is going to need to have its own supply of blood. It cannot use the blood that's moving through the atrium or the ventricle. It's too fast, pressure is too high. There's just not enough time to extract the oxygen and nutrients from them. So it relies on the coronary artery, right? That's on the surface of the heart. So here in this model, they dissolve all the heart muscle and left behind the coronary artery. You can see it's a very extensive network. Now, if the atherosclerosis that we talked about causes a buildup of these fatty material inside one of the coronary arteries, then we're gonna have a problem. Okay? Depending on how much of it is blocked, right? The outcome is the severity of the outcome is gonna vary, right? But you know, in general, we call them coronary artery disease, or CAD. If it's a partially blocked coronary artery, it's not completely blocked, you can still let blood go through, but you might not be able to get enough blood through, right? So that could lead to angina. Angina is chest pain, especially when the person tries to physically exert themselves. Like if they lift something heavy, that requires extra oxygen, right? And the heart muscle needs to pump harder. But if the blood vessels is partially blocked, you may not be able to supply enough oxygen to the muscle in the heart, and that leads to chest pain. If they walk up a flight of stairs, that's like physically exerting themselves, that could brought on the chest pain, okay? Angina. So what do we do for people who suffer from angina? Well, they are usually given aspirin. Aspirin is good for a painkiller, but aspirin actually is also used as a blood thinner. It makes the blood thinner, and when it's thinner, it's able to go through narrow space easier, right? So, you know, if you visit a dentist, sometimes they ask you to fill in like a questionnaire, right, at the beginning if you're a new patient. And one of the things they ask you is, are you on aspirin? Because they have to know. Dental work, there's a lot of bleeding that goes on usually, right? And if you are on aspirin, I mean, aspirin is not the only blood thinner. There are other blood thinner too, like warfarin. You might have heard of that, right? But anyway, the point is, like, if you're on blood thinner, your blood don't clot as easily. So if you're bleeding, you know, you might not be able to stop the bleeding easily, right? So they have to know. There's another drug called nitroglycerin. Nitroglycerin is used as a vasodilator. Okay? So it could be a pill that they put under the tongue. You might have heard of that before. They also have a spray. And it basically opens up all the blood vessels, make them bigger so the blood can pass through. Okay? It's interesting because nitroglycerin was first discovered as an explosive. They actually use this in weapons to make explosives. But somehow they also found out if you ingest this stuff, you know, it opens up your blood vessel. It's interesting, you know, what kind of things some of these compounds can do. So another thing that people can do if they have a partially blocked artery is balloon angioplasty. So in this case, they would insert a catheter. A catheter is like a tube, right? They usually do it up the femoral artery from the leg, okay? And then they would feed the catheter all the way to the blocked area in the heart. They don't just do it like blindly, okay? They have ultrasound to help guide the catheter, okay? And of course, they have to figure out where it is blocked to begin with, right? So there are these dye that they can inject into your blood vessels, and then they could take an X-ray, and we call that like an angiogram, right? So they can figure out exactly where it's blocked. They could do this balloon angioplasty. They feed the catheter up your legs, get to where it's blocked, and then they inflate the tip of the catheter. And the tip of the catheter is a balloon. So basically, it's like this. Okay, like let's say you have a little bit of this block stuff here. So you put the catheter in place, and then you inflate the balloon. So when the balloon gets inflated, it pushes the plaque against the wall and make it flat. And when you pull the balloon out, then the plaque has been flattened, and you reopen up the blood vessel to restore the blood flow, right? So people who do balloon angioplasty, who receive angioplasty, I should say, there is a 25% chance where the blood vessel would eventually narrow again, which makes sense because you're not really removing the rough patch, right? So this is still not smooth. So more blood clot can form, and then eventually it will come back, right? And so what happens is nowadays they could leave like a stent behind, STENT. After they pull the catheter out, they leave a stent. So it basically, the stent basically keeps the blood vessels open and prevents it from closing up. Okay, so that deals with re-stenosis. Remember, stenosis is narrowing, so we prevent it from narrowing again. Questions, anyone? Okay, what else can you do? Well, if it's completely blocked, then it would lead to a heart attack, right? Because if it's blocked, then you are not sending oxygen at all, and that part of the heart muscle can die off. Right? We call that a heart attack or myocardial infarction. If it happens in the brain, then instead of a heart attack, what will the person get?
A stroke.
A stroke, that's right? So equally bad, right? If not worse. So then if the person survives the heart attack, then what they need to do is they have to do this is called a coronary bypass. Is it here somewhere? Here, I'll write out coronary bypass. Okay, so in coronary bypass, they take a vessel from the leg, because you have a lot of redundant ones in the leg. So they take one up, and then they create a graft. Basically, it's like a detour. Right? Hook one end to the aorta, hook the other end somewhere downstream of the blocked area. So let's say this is blocked, right? So now you create the alternate path for the blood to basically flow around the blocked area, right? And then it restores the blood flow. So people might have like double, triple bypass. What that means is you have two of these detours or three of them, depending on how bad the blockade is, right? So I mean, like you cannot do this indefinitely. If the heart is too damaged, then the only other option is to wait for a heart transplant, right? So that's it. That's everything you need to learn from the first lecture. Please work on the study guide. It's very important that you do. Okay, there's a lot of stuff in the study guide that you need to go through. Lots of questions here, fill in the blank, so on and so forth. But there are also application questions. Okay, you definitely should work on the application questions. I will not be giving you the answers to the application questions, but they might show up on your quiz. Right? Not like you have to type out the answer or anything, but then you should maybe do a little bit of research on that. You may discuss it with your classmate. There will be some multiple choice question related to these things on the quiz. Okay, so that is all. Anything else you guys want to ask before we end for this week? The answer will be posted, right, for the study guide. Right? Just not for the application questions. So you can go check them out. Okay, that's it then. If you think of anything else, just email me. Otherwise, I will see you next Tuesday. Have a good one, guys. Bye-bye.
Thank you.
Thank you.
Professor.